Hi there, good afternoon. So in this lecture, I'm going to go ahead and cover Chapter 19, Major Pathogenic Gram-Positive Bacteria. And I'll go ahead and cover several categories, several genuses of the Gram-positives. Just real quick, these are, first of all, these are bacteria that cause disease in humans. Okay, that's where pathogenic comes from. And then Gram-positive comes from the gram stain, the technique that we learned early on in the semester that allows us to stain the cell walls of bacteria. Recall that Gram-positive stain purple. Okay, so these are the different genuses that I'm gonna go ahead and cover in this lecture with regards to their structure, their physiology, pathogenicity, and a little bit about their epidemiology. Okay, so starting off with Staphylococcus, Streptococcus, Enterococcus, Bacillus, Clostridium, Listeria, Mycoplasmas, Corynebacterium, Mycobacterium, Propionibacterium. So I'll go ahead and go over a few of the diseases as well, which I'm actually highlighting here. For example, for Staphylococcus, these are two different specific species of the Staphylococcus genus. So we have Staph aureus and we have Staph epidermidis. This one we've worked with in the lab previously. This is one of the bacteria that you actually grew and you cultured, and you were actually able to gram stain. And if you go back to your notes, you should find it there somewhere, and it should be gram-positive. Okay, so again, gram-positive bacteria, they are gram-positive because they stain purple, meaning that during the gram stain, they don't keep anything on their cell wall except for the crystal violet. Okay, so the decolorizing agent, the alcohol, is not able to remove that completely, and their cell wall is not going to take up safranin, which is the last chemical of the gram stain. So let's go ahead and begin. Okay, so start off with Staphylococcus, which you're looking at here on this diagram. This is bacteria bacteria from a specific colony, which was grown on nutrient agar, which is the background right there. Okay, so this micrograph comes from a scanning electron microscope, and the scale is at 4 micrometers. So first and foremost, they are purple, they're gram-positives, and they're divided up into two major groups based on their DNA composition. Okay, so the groups are, some of them contain low G+C bacteria, meaning that in their genomes, if you were to analyze their genomes, you would find a low quantity, low percentage of guanine and cytosine bound together in their DNA. Okay, the other group is a high G+C bacteria, which is the opposite of the low, meaning that the high G+C would contain a high percentage of guanine cytosine base pairs found in their genome. Okay, so keep in mind, guanine and cytosine are two nucleotides of the of DNA. Okay, so every single living species, every living thing, whether it's bacteria, animals, plants, they all use basically the same DNA, the same 4 nucleotides. The other two being adenine and thymine. Okay, so now what's important about these bacteria is that they form part of our normal microbiota, okay, which means that they live on and in us. Okay, so they're going to be, if you were to swab your own skin, you would probably find Staphylococcus there after a 24 to 48 hour, maybe 72 hour incubation period. And so again, they are normal microbiota, and but they can be opportunistic pathogens given that they find a way to infect, the cells that where they're not supposed to be found. Okay, so if you find them on the skin or maybe in the gut, but for some reason they end up colonizing other parts of the body, like maybe the eyes where they're not supposed to be, maybe the nasal cavity, then they can cause infection. Okay, that's why they're opportunistic. In terms of their structure and a little bit of their physiology, they are facultative anaerobes. Okay, so meaning that they can grow in the absence of oxygen, but they can also grow in the presence of oxygen. So they're pretty tolerant to oxygen. Keep in mind that bacteria use mostly glycolysis and fermentation to be able to produce their AGP. They cannot necessarily do aerobic respiration, but they can still carry on with their metabolism in the presence of oxygenic, reactive species like ROS, for example, or reactive oxygen species. Okay, so now, as you notice here in the diagram, they do grow in grape-like clusters. So their colonies are gonna look like a grape bunch, if you will. They don't move. They're not necessarily motile, so there's no flagella that allow them to move. And they do tolerate a certain amount of salt, like for example the sodium chloride that's found on our skin. So that sodium chloride, which is excreted as a waste product. So if we do have an excess of sodium and chloride inside of our cells, then we manage to excrete it not only through the urine and feces, but also through the skin. Okay, so these bacteria are gonna actually be able to live there. They're gonna be pretty tolerant to it because of their cell wall and other internal structures. They are tolerant of desiccation, radiation, and heat, meaning that they can survive on environmental surfaces with even if the sun is bright and very radiant outside. They won't necessarily dry out because, again, of their thick gram-positive cell wall, and they can they can again be tolerant to radiation as well, so UV radiation. So again, two major species of Staphylococcus bacteria. We are looking at Staph aureus and Staph epidermidis, which do live in an honest, but at times they can be opportunistic as well, meaning that they can cause disease. When they do cause disease, that means that they have breached first line defenses, so physical barriers, and they can do so, they can survive. If they do that, they can survive in the body or in organs where they're not supposed to be in three general ways. So their pathogenicity, if you will, depends on three mechanisms. First, structures that enable it to evade phagocytosis, meaning that they're going to go ahead and stave off attack by our macrophages. Okay, second is they can produce enzymes to do the same thing and prevent the complement system from attacking them. Remember, the complement is a set of proteins that comprises part of our immune system. And then they can also produce toxins to be able to, again, fend off any attack from the immune system. Okay, so first and foremost, in terms of structural defenses against macrophages, is they secrete a few molecules here to be able to do that. Okay, first and foremost is protein A. So what protein A is going to do is it's going to go ahead and help defend against immunoglobulin attack. Immunoglobulins if you recall from the immune system, they are antibodies. Okay, so protein A is going to prevent IgG, which is one of the antibodies, from opsonizing that bacteria. Okay, so what does that mean? That means that the antibody is going to normally, right, it's going to go ahead and produce a coat around the bacterial cells, and in that process present it to the rest of the immune system, to the macrophages, so that the macrophages can eat it, okay, can engulf it. But protein A prevents it from being, opsonized by these antibodies. Okay, second is coagulase. So coagulase is an enzyme that it plays a role in the prevention of clotting. Okay, so what this is going to do is it's going to go ahead and convert something called fibrinogen into fibrin molecules, which is part of the clotting, blood clotting mechanism. Okay, so what this is going to do is the bacteria is going to go ahead and use fibrin and clots, for that matter, as some sort of a cloak, right? So it's going to go ahead and provide, metaphorically, obviously, invisibility, if you will, to the immune system. And in that manner, they can essentially hide from the immune system. Okay, so pretty smart bacteria there. And then last but not least is formation of slime layers in capsules. Capsules form around the bacterium. So what that's going to do is it's going to go ahead and inhibit leukocytes from carrying on with their chemotaxis. If you recall, chemotaxis is the signaling pathway that our chemokines use to be able to call on the immune system to go ahead and mount an entire attack against bacteria. Okay, so these Staphylococcus bacteria are going to go ahead and prevent that by inhibiting leukocytes. And the slime layer is also going to help them in attaching to surfaces. Okay, so second aspect of their pathogenicity is the use of other enzymes. Okay, so for example, cell-free coagulase, which is going to trigger blood clotting, and in this case, it's important that they do trigger some sort of blood clotting, so that they can use fibrin as that quote-unquote invisibility cloak, if you will. Next is hyaluronidase. So this is an enzyme that breaks down hyaluronic acid, which is found on the skin, and I'll show you some pictures right now of what happens when they do produce that hyaluronidase, and it acts against our skin epithelium. And last but not least is staphylokinase, which is gonna go ahead and dissolve fibrin threads in blood clots to allow the bacterium to free itself once it's inside of an organ that it must attack. Okay, so here's an example of the use of hyaluronidase on a patient with scalded skin syndrome. So hyaluronic acid is a normal molecule that we have that our cells express, our skin cells express, to be able to keep the epithelial cells, the squamous epithelium cells together. So hyaluronidase breaks that down, which allows for these cells to spread out, and the bacteria can actually grow right in between the cells. And you're seeing the detachment of skin cells because of the use of hyaluronidase in bacteria. So another enzyme that helps them survive on oily skin is lipases. Okay, lipases break down lipids, and so we do secrete lipids onto our skin to be able to reflect UV radiation off of our skin and provide and keep moisture inside of our bodies. So these bacteria and their production of lipases will digest those lipids, and therefore allow for the bacteria to grow on skin. even oily skin, and also within oil glands. So beta-lactamase is another enzyme that they do produce, and beta-lactamase allows them to survive attack by antibiotics. In terms of toxins, there's a few toxins here which mainly disrupt cells, some cellular mechanism, for example the membrane, right? So the integrity of the membrane can be disrupted by toxins that these bacteria produce. So staph bacteria will produce a nice set of toxins here. For example, cytolytic toxins that disrupt membrane, exfoliative toxins which are gonna cause skin to slough off, such as you're seeing here, and then toxic shock syndrome toxins as well, which I'll go into in a little bit, and then enterotoxins, which are mainly molecules that disrupt the gut. Okay, so they're gonna go ahead and allow this bacteria to colonize the gut and cause gastrointestinal diseases, like for example food poisoning. So the TSS toxin, which stands for the toxic shock syndrome toxin, is produced by a staphylococcal species, which can grow not only on the skin but also on surfaces, for example, of tampons. Okay, so these are reproductive products which, if left inside of the vaginal canal for longer than is necessary, then this bacteria can actually grow on it and cause percolation of this bacteria into the blood, which can then travel throughout the body, and since they're non-motile, they can the blood moves them, and they can stay stuck in tissues and grow on tissues, such as you're seeing here on this woman's arm. So TSS does cause a set of symptoms, like for example rash, which you're looking at here, fever, and it can lower blood pressure as well to dangerous degrees. So now, if left untreated, then toxic shock syndrome will cause disease. So the FDA required labeling of tampon boxes, and ever since the 80s, so this happened in the 80s, the incidences of toxic shock syndrome have been lowering. So here we are in the 80s, right, where you see where we saw a peak in the incidences of TSS, but then after labeling, you're looking at a decrease, progressive decrease in toxic shock syndrome incidence there. And it can cause death if left untreated, not only because of TSS, but also because the bacterium can become attached to other tissues and cause different diseases. For example, bacteremia, which is the presence of bacteria in the blood, which is not supposed to be there. We're not supposed to have bacteria in the blood, so it's supposed to be sterile, and normally it is. Next is endocarditis. So this bacterium can also dock and attach on the endothelial surfaces of the heart, which is basically the inside lining of the heart, of the chambers. So if it does that, then it's going to cause inflammation and then tissue damage inside of the heart, which can evidently be fatal. Next is pneumonia. Pneumonia is an infection of the lungs, And last but not least is osteomyelitis, which is an infection of the bones and bone marrow. So obviously prevention is of utmost importance, and that cleanliness here becomes key. But if infection is found, what the doctor is going to do is she's going to go ahead and order lab diagnostic tests, and lab technicians are going to look for the grape-like growth colonies, and if that is found, then the treatment is going to consist of methicillin and vancomycin, due to the fact that a lot of species of the genus here are resistant to other antibiotics like penicillin, for example. Okay, so next is the Streptococcus genus, which are also facultative anaerobes, and the difference is that their bacteria are cocci, which are arranged in pairs, and they can even be found in chains under the microscope. Okay, they are again gram-positives, as mentioned, and they can be categorized into two large groups A and B, with many human pathogens here. So what we're looking at here is a Streptococci bacterium colonizing the throat. Okay, so we're looking at pus pockets here on the tonsils, and what that is basically the bacterium being attacked by the immune system. So these are white blood cells, leukocytes, phagocytes, monocytes, which are attacking the bacteria. Okay, so that's a disease called pharyngitis. The tonsils are comprised of immune tissue called MALT, if you recall from the immune system lecture. So again, Streptococcus, let's go over group A Streptococcus, starting off with Streptococcus pyogenes, okay, which is a specific species of strep. Its pathogenicity is gonna depend on a few characteristics here. First is trying to evade phagocytosis, so evading those, staving off those immune system cells that engulf and eat bacteria, like macrophages, for example, and complement system. So what the bacteria is going to produce is this M protein to essentially destabilize the immune system, specifically the complement proteins. So that's against the immune system, evidently, but the bacteria is also going to produce other molecules which are going to allow the bacteria to spread out through the tissues. Okay, so one of which is called hyaluronic acid, streptokinases is another one, and deoxyribonucleases. Again, these three are going to help the bacteria spread out throughout the tissues and form colonies like you're seeing here in the throat of this person. So specific proteins and enzymes that are produced by this bacteria include C5a peptidase, hyaluronidase, pyrogenic toxins, and streptolysins. So starting with the peptidase, C5a is a complement protein. So peptidase is going to go ahead and break down C5a, inhibiting many of the main mechanisms and pathways through which the complement system works into. If you can go back to the lecture in the notes on the complement system, you'll be able to note how C5a is involved in lysing bacteria, okay, and attacking viruses for that matter, and opsonization, neutralization of toxins as well. So again, breaking down that protein prevents the immune system from attacking the bacteria. Hyaluronidase, which is very similar to what the Staphylococcus genus does, it breaks down hyaluronic acid, which prevents the cells from attaching, so it detaches cells basically. Okay, and that's used to be able to spread out throughout the tissues and form colonies. Pyrogenic toxins are gonna stimulate fever, rash, and shock in the person. And streptolysis are gonna work by inhibiting and destructing erythrocytes or red blood cells of the patient. Not only red blood cells but also white blood cells. So streptolysis would inhibit the action of white blood cells on the bacteria. But streptolysis also cause depletion of platelets and their formation from megakaryocytes. So Streptococcus again can cause a range of diseases, mainly from not treating the main disease, which is caused by strep pyogenes, and we're looking at it. Right? This is pharyngitis again. So not treating this disease can cause the bacteria to filter to the blood through the tissues, percolate to the blood, and spread to other parts of the body. So from pharyngitis, people can end up with rheumatic fever, which can damage heart valves and the muscle of the heart. It can also cause autoimmune responses to heart tissue, which can obviously be pretty dangerous for the person and even cause disease. Or cause death, I'm sorry. Now, scarlet fever is another complication of not treating pharyngitis. So what happens here is a chest rash, okay, so which not only develops on the chest, but can also spread throughout the rest of the body. But again, those are extreme cases due to not treating pharyngitis. So other diseases that strep can cause are erysipelas, which you're looking at here. These are pus-filled lesions on the skin. But there's also strep toxic shock syndrome. So not only staph can cause TSS, but also strep. Okay, so that happens when, again, blood or the bacteria percolates into the blood, and the blood moves it throughout the tissues, throughout the body. Okay, which can cause organ failure, can make the person go into shock, and even death because of the shock, which mainly affects the cardiovascular system. So this can also cause necrotizing fasciitis, which essentially means destruction of the underlying tissue of the skin. Okay, so if the bacteria manages to get through the skin because of the use of hyaluronidase, then they can penetrate to the underlying tissue of the skin, which is made up of fat and connective tissue. So bacteria love connective tissue under the skin, that bacteria, and they start eating through it to produce, again, necrotizing fasciitis. And necrotizing, it means dead tissue, essentially. So this bacteria, if it does percolate to the blood, can cause, again, organ failure. And one of the organs that are mainly attacked here are the kidneys. Okay, within kidneys, we have 3 different layers in the kidney: one superficial, one middle, one deep layer. The superficial is called the kidney, the cortex of the kidney. The middle is called the medulla. The deep layer is called the pelvis, or the renal pelvis. So within the cortex, and to a certain extent within the medulla, we have glomeruli. Glomeruli are part of nephrons. Okay, each of our kidneys contains about 1 million nephrons, which are anatomical structures that filter blood and produce urine. So the way in which they filter blood is by using glomeruli, okay, or glomerulus for one. Okay, so those are spherical structures that through which blood vessels penetrate. So they go ahead and penetrate through the glomeruli. They have an afferent arteriole that goes in, and then the blood vessel essentially forms a network inside of the spherical glomerulus, and then has an exit arteriole, which is called the efferent arteriole, that comes out of the glomerulus. So again, bacteria moving into the blood manages to attack the glomerulus and cause colonies and infection of the glomerulus. So that's called glomerulonephritis. Okay, so what does that cause? It causes inflammation of the kidneys, which means low urine output, and even bacteria in the urine. Now, the diagnosis of strep pyogenes would come first through a lab test. So these lab tests have to be able to find these gram-positive bacteria that are formed in chains or in pairs. Okay, and these are cocci spherical bacteria that are again paired, or they can form chains as well. There is a rapid strep test that can be used, and so this for this strep test, the doctor needs to basically get a sample, right? So a throat swab essentially, and then the treatment is gonna be comprised of penicillin. So if there is no resistance, then the patient's going to go ahead and be prescribed penicillin, and or allergic reactions for that matter. But also, I guess the best way, right, to prevent infection of this bacterium is cleanliness. Yeah, so not only cleanliness, washing your hands and whatnot, but also having a good diet in order to cause the generation of antibodies that can kill and prevent, for that matter, entrance of this bacteria into the blood. Alrighty, so now on to the group B Streptococcus genus. So these are differentiated from the group A strep in the fact that these form cocci, or these are cocci shaped that form chains exclusively. Okay, so any diagnostic test for a disease caused by group B Streptococcus would initially be looking for these chains, okay, cocci chains. So now another way to differentiate them from group A Streptococcus is by beta hemolysis or hemolysis. Hemolysis refers to the fact that these bacteria like to break down erythrocytes. Okay, they break down red blood cells. So if you were to grow them on blood agar, if you were to culture them, you would use blood agar, and you would essentially carry on with their identification in a lab by looking for two patterns. Okay, so you'd be looking for alpha hemolysis, which is essentially bacteria that do grow on blood agar and that break down blood, and you would see that in a color change which looks greenish. Now what that signifies is that even though they do break down erythrocytes, they break them down partially. Okay, so that would be alpha hemolysis, whereas beta hemolysis is a complete breakdown of red blood cells, which appears yellowish in the petri dish. All right, so there's no need to see the color changes under the microscope, you can see them. You can see it right on the petri dish itself, because the bacteria would leave or will leave zones that are, visible to the naked eye. So group B streptococci bacteria like to infect the gastrointestinal tract. Okay, starting in the mouth, as you're looking at here in this diagram. So the GI tract starts in the mouth, continues to the oropharynx, to laryngopharynx, and then down into the esophagus, stomach, small intestine with its 3 divisions, the duodenum, jejunum, and the ileum, and then the large intestine with its 4 major divisions, or 5 major divisions rather, ascending colon, transverse colon, descending colon, sigmoid colon, rectum, and which includes the anus, rectum and anus. So again, group B streptococcus bacteria are mostly gastrointestinal disease causing bacteria, but they can also be opportunistic in the genitals and the urinary tracts. Okay, so this specific species that's causing dental caries is called Streptococcus mutans, M-U-T-A-N-S. But let me go over the pathogenicity here real quick and general characteristics of the group B before I get into more details on this. So in terms of pathogenicity, these bacteria are generally gonna go ahead and infect newborn babies because of the fact that they don't necessarily have the antibodies to kill off these bacteria. Okay, so as the baby's coming out of the vaginal canal, the mother is gonna basically inoculate the baby with this bacteria, and throughout the age progression of this baby, he or she is going to go ahead and develop antibodies as they go on. But again, in some babies that don't necessarily have good amounts of antibodies, then they can basically be born ill, or maybe streptococcal disease, soon after birth, given that they don't necessarily have the antibodies. Regardless, it can be treated with antibiotics, and I'll go over that in a little bit. So diseases that are associated with these bacteria are mainly neonatal bacteremia, meningitis, and pneumonia. But again, in some species of it can cause dental caries as well. So in terms of diagnosing, the lab technician will need to use an ELISA test. If you recall, ELISA stands for enzyme-linked immunosorbent assay, which uses bacterial toxins in the identification of disease in a certain sample. The treatment of any disease caused by a Streptococcus bacteria is gonna depend on penicillin administration or ampicillin administration, depending on whether the patient is allergic to penicillin. But penicillin can be administered as a prophylactic drug right after child birth to evidently reduce the incidence of infection in newborn babies. And then women who get immunized for or get vaccines for streptococcal bacteria, they can pass on those immune cells onto the baby. So here we're looking at Streptococcus pneumonia, which is an opportunistic bacteria that infects the lungs of people that are already immunocompromised. So if you are anyone happens to get the common cold, which is caused by a virus, this bacteria, that person becomes immunocompromised within their pulmonary system. And given the existence of this bacteria within either their surroundings, or maybe in some way they became exposed to it, which is pretty simple to do, then this bacteria can become pneumonia, right? Which is evidently it can be a fatal disease, which infects the lower respiratory tract of a patient. The lower respiratory tract consists of the lungs and everything that's within the lungs, to include the bottom portion of the trachea, which is known as the carina. So the carina bifurcates into primary bronchi, which divide up into secondary and tertiary bronchi, bronchioles leading all the way to air sacs, anatomically known as alveoli. Okay, so those alveoli contain a set of cells called type 1 and type 2 cells that again can become infected by this bacteria right here. So the specific phenotypical characteristics of this bacteria that allow it to cause disease, so its pathogenicity, if you will, is due in large part to its capsule. Alright, so this capsule is made up of polysaccharides, which basically make the capsule that bacterium very strong, so very strong and resistant to any environment that might kill it. Okay, so it's a very thick, essentially very thick cell wall made up of polysaccharides, which are carbohydrates, that again give it its resilient, if you will, characteristics within the body. So it also contains and secretes some molecules, for example adhesin, which allow this capsule to bind on to the cells of our tissues, at least within our respiratory system, beginning in the pharynx all the way down to the trachea, bronchi, bronchioles, alveoli. It also secretes IgA protease. IgA is immunoglobulin A, which is an antibody. So protease is an enzyme that Strep pneumoniae releases to kill the antibody. Okay, so if it kills the antibody, right, you can imagine that this bacteria can colonize. It has more of an opportunity to colonize the pulmonary tissue. So it also secretes something called pneumolysin, and this pneumolysin destroys epithelial cells. Epithelial cells are in case you're not familiar with epithelial, it's basically the outside covering of any body system that's exposed. Like for example, the oral cavity, trachea, and bronchi, they're exposed to the environment. Yeah, so they're open to the environment. And so again, another characteristic of this bacteria is the production of pneumolysin, which lyses epithelial cells. Okay, so in that process, the bacteria can colonize, pulmonary tissue in that manner. So other diseases that this opportunistic pathogen can cause in our body are sinusitis, otitis media, which are diseases of the sinus. That would be sinusitis. We have several sinuses in our facial that are made by our facial bones. So this bacteria can infect the sinuses. It can cause otitis media, which is an infection within the ears. Yeah, so the middle to inner ear. It can cause bacteremia if it does percolate into the blood. Endocarditis, right? So again, percolation into the blood of this bacteria releases, and the fact that it releases toxins means that it can infect the blood. It can dock on the internal tissue of the heart, the endothelial cells of the heart, to again cause inflammation of the heart and destruction of heart tissue. And another one would be meningitis. So meningitis can be caused by different types of bacteria and even some viruses. But pneumococcal meningitis has a really high mortality rate. So if you're not familiar with the meninges are the outside covering of the brain. There's about 3 to 4 layers, very thin layers, that allow for something called CSF, cerebrospinal fluid. Cerebrospinal fluid contains many substances to include glucose. It contains immune system cells, among many other molecules, nutrients and the like. So this bacteria can make it all the way into the meninges and release its toxins to cause the disease known as meningitis. So the diagnosis of this specific bacterial strain within the Streptococcus genus would be done by 1 gram stain. Evidently, it's a gram positive, and 2 is alpha hemolysis on blood agar. The treatment consists of penicillin, although some resistant strains have emerged. So there's current work on other antibiotics of choice for the specific patient. But there is a preventative vaccine which is made from the capsule material of the bacteria. Okay, so that's that for Streptococcus. Alright, so let's get into the Enterococcus genus of bacteria. We're looking at Enterococcus faecalis right here. Enterococcus bacteria are found mainly within the gastrointestinal, or the large intestine rather. Okay, so these live normally within the large intestine. There's two species of importance. One is faecalis and one is faecium. So these are healthcare related, medically important bacteria. So what they're gonna do is they're gonna be found in short chains and pairs. So they do live in the colon and help humans in producing feces, but they're also involved in the production of vitamins. Again, vitamins that come from the gut, from the large intestine. So, given the fact that they live in the human colon, they don't necessarily cause disease there, but they can become pathogenic if they do come in contact with any other part of the body. So this is very common in early childhood, in babies, and also throughout the toddler years, given the fact that for the most part in early developmental, kids are not necessarily potty trained. So they're touching their their rear constantly, and not necessarily washing their hands effectively. And even in early potty training, kids sometimes forget to wash their hands, and they're touching parts all over their body, their face, their eyes, putting their hands in their mouth and whatnot, picking their nose. So therefore, if their hands are infected with Enterococcus faecalis, for example, excuse me, having a little bit of difficulty here, and they bring that infected hand over to the eye, for example, then it can cause infection in the internal aspect of the eyelids. So again, not pathogenic within the gut, but they do cause disease outside of the gut. So diagnosing this or infection with this bacterium does take a gram stain. So you would be looking for the gram-positive aspect of these bacteria, and then you'll also be looking for the shape of the bacteria, so the arrangement that they produce. And the treatment of this bacteria would consist of antimicrobials, but it's difficult to a certain extent because they are resistant to a wide range of antimicrobials, to antibiotics. Okay, so the administration of these antibiotics is going to have to be at higher doses and for prolonged periods of time. So instead of, say for example, the patient was supposed to take the penicillin maybe once a day, right? 2 pills once a day. The resistant nature of the bacteria is going to take a lot more exposure to the antibiotic, thus forcing the doctor to prescribe antibiotics for a prolonged period of time, so higher doses several times a day. Okay, so they would, for the most part, antibiotic treatment lasts about anywhere from 10 to 15 days. So in these cases, and especially in immunocompromised patients, they would have to, prescribe those antibiotics for over 15 days, sometimes even up to 4 weeks. So the fact that they are difficult to treat and pretty easy to spread, that calls for evidently preventative measures. And the most important preventative measure that we can teach kiddos is evidently wash hands, and wash hands effectively, warm water, and or warm to hot water, and soap. Alrighty, so the next genus is the Bacillus genus. We're looking at Bacillus anthracis here, which is the causative agent of anthrax. Okay, so these do produce an anthrax toxin. They are facultative anaerobes, and they are bacilli in shape, or rod in shape. You can actually see them right there in this scanning electron micrograph, magnified to 10 micrometers here. So you can see the rods, right? So these are called bacilli. So these are going to form chains, right? So any diagnosis of Bacillus anthracis in any Bacillus genus species would basically look for these, for these bacilli rods. These bacteria are unique in the fact that they do produce endospores, and the production of endospores allows this bacteria to be highly adapted to many different types of environments. Endospores, if you recall from early in the semester, endospores are excuse me, small little capsules that contain the DNA of the bacterium, which are highly protected by a peptido or a carbohydrate capsule, which basically give the bacteria the skills, adaptations necessary to be able to maintain a dormant period in case they're not in a beneficial environment. So if the environment does not favor their growth, then they go ahead and become endospores, become dormant until they are in a beneficial environment. So outside the body, these bacteria are going to go ahead and live in aerobic form. The endospore form is going to be transmitted in one of three routes. So first is inhalation, right? So you can breathe in the spores. You can have inoculation of spores through a break in the skin. So any type of injury, any open wound that becomes exposed to the endospores of Bacillus anthracis would basically, inoculate, make you sick. And last but not least is ingestion, so by eating these endospores. Okay, so we're looking at a disease here caused by anthrax. This is cutaneous anthrax, which causes these very dark lesions and bumps, for that matter, on the skin. Cutaneous means skin. Okay, so this is basically one way in which one can get sick, one can get infected with Bacillus anthracis. And these lesions are called ulcers, which are, fatal if left untreated. All right, so they don't only happen. I know I'm showing you here the fingers, the hand, but they can occur anywhere on the skin of the patient. And cutaneous is just one way of getting infected with this bacterium. Another way is inhalation, right, of anthrax, of the endospore anthrax. So if somebody does manage to inhale it, then the bacterium, the endospore rather, will actually become a bacterium. It'll divide within the lungs, within the pulmonary system, respiratory tracts, upper and lower, and this is going to have a really, really high mortality rate. So last but not least is GI anthrax. So this is, even though this is highly rare, this is very rare in people, it is important that you know that there is possibility of anyone becoming infected with anthrax by eating. So these bacteria don't necessarily move. So essentially, wherever the endospore lands, once it manages to penetrate the body, then, or our first lines of defense, then they go ahead and stay non-motile and start dividing right there in that part of the body. So the diagnosis would be, one, they're non-motile, so they won't necessarily move. They are gram-positive bacilli, so you would be looking for that purple color. And the sample would come from either the lungs or the skin. So treatment for Bacillus anthracis would be comprised of antimicrobial. So any wide range or broad range antimicrobial would be effective against Bacillus anthracis. This evidently if treated within a certain time frame. Before it causes necrotizing, for example in cutaneous disease here, if it does manage to cause necrotizing ulcers, then that ulcer right there is pretty much, or that piece of skin is pretty much gone at that stage. So and as long as the disease is caught in time, and the patient manages to treat it within a certain period of time, then there's not a lot of issue there. So in preventing anthrax, there is a vaccine. Okay, so this vaccine would require boosters. So one main vaccine, and then boosters after the first administration. All right, so moving on to Clostridium genus, we are looking at a disease called gangrene, or an infection caused by Clostridium perfringens. Okay, so this disease is commonly known as gas As the bacterium feeds on the tissue, it produces a very stinky gas. Okay, so a very unique smell comes out of the metabolism of this tissue, and you are basically looking at the bacteria eat right through the tissue. Evidently, you can't see the gas coming out, but you would basically identify this by the unique smell that the bacterium produces as it eats right through the cells here. So I guess again, the gas that they produce, is the reason why this bacteria is called gas gangrene. So the Clostridium genus or bacteria within the Clostridium genus are mostly anaerobic bacteria that form endospores. Okay, so these endospores, again, as mentioned, they are going to give the bacteria the ability and the adaptation to live under harsh environments, okay, which are for the most part going to be found in soils, untreated water, and then within the intestinal tracts of animals and even humans. So one of the reasons why feces has some sort of smell is in part due to the presence of Clostridium genus bacteria. Aside from many other millions of different species of bacteria, which are all metabolizing waste products that we're dumping right into the large intestine. So their metabolism causes the production of these gases. So in diagnosing Clostridium perfringens, you would be looking for first the gram stain, right, the gram-positive nature of it. You would be looking for bacteria that are non-motile, and then also the presence of minimum bacterial loads in food and feces. So you would get your sample from essentially the feces, and also in cases of gas gangrene, you would basically get a sample, right, of the necrotized tissue that you are looking at in your patient there. So the appearance of gas gangrene is also diagnostic. Again, it does have a putrid smell, and in cases of food poisoning, because these bacteria can also cause food poisoning, then you would basically get a sample of vomit, right, of the patient. So in treating, for example, gas gangrene, what you would need to do is you would need to remove all of this dead tissue right there. Okay, so all that dead tissue, and then hope that the bacterium hasn't made it into the nerves, right? Because if they do, they can cause all sorts of issues with the movement, right? With the movement of the body parts. So as it is, this patient right here will, for sure lose all of this tissue right there, because all of it is going to be removed in order to save the rest of the foot. And that tissue, even though it's scarce, it doesn't grow back. Okay, so the patient is going to be left without all of that superficial tissue there. Again, hoping that the bacterium doesn't make it to the nerves. If it does make it down to the nerves, then the patient would lose two aspects, right? So they're going to lose the motor aspect of the toes, in this case. Motor meaning movement. So they're not going to be able to move the toes, so they're going to have limited movement of the toes. And they can also lose sensory. Yeah, because we have two modalities to the nervous system, sensory and motor. So if those nerves are affected by the toxins of the bacteria, then again, sensory and motor functions can be lost. Alright, so preventing this disease or infection with this bacterium here would basically require that you have a relatively clean space within the home, especially areas that have soils or soils that can act as good places for the endospores of Clostridium to survive in, right? So maybe isolating those places and wearing shoes, right? Basically every time that you're gonna go on to walk outside, and especially on top of a good soil for planting, for example, that soil contains a wide range of bacteria that you would not imagine. And last but not least is in caring for example, this patient, right? And the doctor would surgically remove all of this tissue here, and then after that it's all the work of the nurse, right? So it's just cleaning, caring for what is left of the foot tissue there, and maintaining a rather sterile environment. Alright, so moving on to another bacterium of importance here, Clostridium botulinum. Right, so this bacterium causes the well-known disease botulism. So this bacteria likes to infect the neuromuscular junction, the nerves and muscles. And what I'm showing here again is the junction between the nerve and the muscle, right? So the bacterium is going to go ahead and have an effect right around there. So in normal muscle contraction, what happens here is that at the end of the neuron here, keep in mind this neuron comes from the spinal cord, right? And then it's indirectly connected to neurons of the cerebellum, which are in charge of skeletal muscle contraction. So the nerve ending, the axon terminal of this neuron, would basically release these vesicles, right? If you recall from biology, vesicles are membrane sacs that contain proteins. Those proteins, if they're found in the brain or the nervous system, they're called neurotransmitters. Neurotransmitters allow us to do basically everything that we do. They govern every single action that we embark on. All right. So, for example, the neurotransmitter called acetylcholine is required for muscle contraction. So, if you must contract your muscle to move, which you pretty much have to, then you're gonna require high amounts of acetylcholine to be released into the synaptic cleft or the neuromuscular junction in this case. Acetylcholine is going to bind to its receptors on the membrane of this muscle, and that's going to cause an action potential to be transmitted over to the muscle, which ultimately causes contraction. Right, so if you have taken anatomy here, I highly suggest going back to anatomy 1 and reviewing this very interesting process here. If you haven't taken anatomy, then I suggest you look up at least some readings of a video on how this takes place. Very interesting stuff. So the presence of the botulinum toxin here is gonna block attachment of these vesicles onto the surface, inferior surface of these neurons, okay, of the axon terminal. So that blocking consequently prevent acetylcholine from being exported out to the synaptic cleft right here, thus not allowing the transmission of an action potential to the muscle here. So therefore, the effect of this is going to be a prevention of muscle contraction. So in diagnosing botulism, then you would basically look for the symptoms. Okay, symptoms are paralysis, right? Paralysis of skeletal muscle. And treating this would take antimicrobial drugs, antibiotics, and also neutralizing antibodies. Okay, so in preventing this, it is said to can food properly, and then in kids that are, or infants rather, that are up to 1 year old, then they shouldn't consume any honey at all, and then evidently keeping them safe from anything that can expose them to the bacteria. Okay, so Listeria is the next genus here. Listeria are another kind of bacillus or rod-shaped bacterium, but difference is that this one does not produce endospores. Okay, it is found in soils, in water animals, and it can enter the body by food that is contaminated, or maybe drinks that are contaminated, fluids that are contaminated with Listeria. So these bacteria are intracellular. So you're looking at a bacterium basically punching through through the plasma membrane of the cell here, and then that causing the entry of the bacterium into the other cell through the use of the plasma membrane of the whole cell right here. Okay, so it's going to go ahead and cause its own exocytosis. Exocytosis meaning its own export, and then in exporting itself, it uses a piece of the plasma membrane of the cell to be able to be transferred into, into the next cell. Okay, so that's basically the only way in which it can cause disease once it's inside of the cell. Again, it is an intracellular pathogen. So once inside of the cell, it gets rid of the vesicle that it formed by the exocytosis, and it's able to grow and divide within the infected cell. So the way in which it survives inside of the cell is by producing listeriolysin. Right? It also produces some virulence factors that allow the bacteria to basically deactivate any enzyme or any lysosome that wants to engulf it and eat it. Okay, so it is highly virulent. It can cause a whole range of diseases, to include meningitis. Again, meningitis is not necessarily only caused by one type of bacterial species or viruses. It can be caused by many different pathogens that manage to enter the brain and dock onto the exterior layers of the brain, called the meninges. Diagnosis of this pathogen requires a CSF sample. CSF stands for cerebrospinal fluid. So through a spinal tap, if you will. So the spinal tap will essentially use an injection, right? That is really long. It's got a long needle, long enough to reach to the inside, right, to the inner part of the spinal cord, where it has a canal. So that canal contains cerebrospinal fluid, and given the fact that this bacterium does make it into the brain in extreme cases, then you can find the toxins within the cerebrospinal fluid. So the treatment of it would take ampicillin or any other broad range antibiotic in case the person is allergic, and prevention of it is basically avoiding foods that might contain the bacterium. So food maybe that is purchased out in the street, that is not necessarily cooked to standard, then you might want to avoid that. Also undercooked foods. Okay, so the next genus is the Mycoplasma genus. We're looking at Mycoplasma pneumoniae right here in this scanning electron micrograph, magnified to 2.5 micrometer magnification here. So what these microorganisms are going to do is, well, first of all, they're one of the smallest microorganisms that can colonize the human body and act as pathogens. But once they do colonize, then they will attach to the epithelial cells of the lining of the respiratory tract. Okay? So they'll attach to the stratified squamous epithelium of the nasal cavity, and then moving towards the back of the nasal cavity, the nasopharynx, they'll colonize the pseudostratified ciliated columnar epithelium, which produces mucus, right? And so that epithelium there contains goblet cells. Goblet cells are specialized types of cells that produce mucus. Now, from the posterior side of the nasal cavity all the way down to the bronchioles, we contain some degree of pseudostratified columnar ciliated epithelial cells with goblet cells. Okay, so mycoplasma likes to invade those cells. So if that does happen, then those cells are going to be prevented from producing as much mucus as they normally do, therefore allowing the bacterium to colonize, okay, and produce these colonies here. So the detection and diagnosis of Mycoplasma pneumoniae will be pretty difficult, because these bacterium here are going to require a sample from the lower tract of the respiratory system. That's one. And second is, if you do manage to culture them, it's going to take a long time for them to be cultured. The treatment of it, so therefore the doctor is going to require, so symptoms. Doctor's going to go with the symptoms of it, and then they're going to try and prescribe macrolides, doxycycline, or fluoroquinolones to be able to treat this infection. For the most part, that works. In extreme cases, which are pretty rare, then you would require hospitalization of those patients. So in preventing this, you're going to be required to, practice good hand hygiene, avoid aerosols, contaminated objects, which are also known as fomites, and be careful, with the distancing, right? Because if people, cough it out, if somebody is infected with it and they cough it out, then you would basically catch one of those droplets and become sick with Mycoplasma. So Gram-positive is the next genus of gram-positive bacteria. They are non-endospore forming bacteria that form V shapes. As you're seeing here in this picture, they form V shapes in cultures, or palisades, which are an aggregation of several V shapes. So they are gram-positive, as you're looking at here in this diagram, and they divide by evidently division, a type of bacterial division called snapping division, and they do colonize the skin, respiratory tract, the GI tract, the urinary tract, and the genitals. So this is one of the main symptoms of a disease caused by Corynebacterium. It's called diphtheria. So this disease is caused by a species called Corynebacterium diphtheriae, and it is characterized by the formation of the pseudomembrane in the back of the oral cavity, or the oropharynx. But keep in mind that at that point in the oropharynx, that is connected to the nasopharynx. So in that area, that's where the pseudomembrane is produced in diphtheria. Okay, so at this point, this bacterium that produced the pseudomembrane released the toxin, and that's the only way in which the disease can manifest symptoms. Because if the bacterium does not release the toxin, then no symptoms are going to be seen on a patient. Now, the reason why this is important is because this is characteristic of diphtheria, but it's pretty rare in the US. It happens mostly in parts of the world that are very, very poor. So again, if it does happen, then the doctor would be looking for the pseudomembrane, formation of pseudomembrane, and that is indicative of diphtheria right there. So treatment of it will take administration of toxins antitoxins that are going to inhibit the toxin of the bacteria. In extreme cases, which are pretty rare, if there is the blockage of the airways here, then surgical removal of the pseudomembrane and opening of the airways is going to be required. But for the most part, people would get better with penicillin, erythromycin in case the person is allergic to penicillin. There is a vaccine, and I believe every single person in the US is required to get that vaccination against diphtheria. Alright, so now mycobacteria is another genus of the gram positives. This is another non-endospore forming pathogen, and it is characterized by the fact that it produces mycolic acid. Okay, so this mycolic acid would be found on the cell cell wall of the bacterium, and mycolic acid is going to allow this bacterium to grow slowly but surely. It's going to protect it from lysis after phagocytosis. So if macrophages do engulf these bacteria, they're essentially going to kill the macrophage, even though they're inside of the macrophage, if that makes any sense. So they make it explode once they're inside. Alright, so now they do have the capability of growing inside of cells, so it would be an intracellular pathogen. And the fact that they have mycolic acid allows them to not accept either crystal violet or safranin. Therefore, gram stain would not necessarily work in many cases, although you're looking at them here. Now, Mycobacterium contains many different species, one of which is healthcare, has healthcare significance here. Tuberculosis is pretty common, although it's not necessarily virulent or deadly. Yeah, so there's several different prevalences here, but first of all, it is a respiratory disease, right? It's caused by the species Mycobacterium tuberculosis, and again, it's not necessarily highly virulent. So there's three major types. First is we have primary tuberculosis, which is basically the initial infection with the bacterium. Okay, so second is the secondary tuberculosis, which happens after initial infection. So if somebody gets it the first time, they're going to go ahead and get sick. The first time around, symptoms are going to be highlighted evidently because it's the first time the person gets sick. But then the second time around, the re-establishment, if you will, of the infection after a secondary activation wouldn't necessarily be that bad for the person. So the person would not show a lot of symptoms, and if they do, they won't be sick for a long period of time, at least not as long as the first time. Now the next one, the third one, is called disseminated tuberculosis, and this results when infection spreads throughout the body. All right, so the bacterium not being treated with antibiotics. All right, so we're looking at a primary infection of tuberculosis. This primary infection is shown here. It happens within the alveoli of the lungs. Okay, this is a respiratory infection here. So that's where it begins, right? Within the alveoli, as mentioned before, we have type 1 and type 2 alveolar cells, along with alveolar macrophages. So tuberculosis actually attacks macrophages and they go ahead and divide within macrophages, making them explode, which exposes them to other macrophages after the initial infection of macrophages. So these new macrophages are also going to get infected. They're also going to get killed at the same time, culturing the bacteria, releasing the bacteria, and starting the cycle again. So that's how these bacteria proliferate. And in case there's no treatment, right, in case it's not treated, then it goes ahead and spreads, it disseminates to other parts of the body, some of which are more clinically important here, like for example the spleen. We're looking at tuberculosis here in the skin, in the spleen, the blood, lymph nodes, bone marrow, spleen, kidneys, spinal cord, brain. So again, these are rare cases here, and here in the United States we don't have a lot of cases, but if you do see some cases, then you're gonna go ahead and deal with the symptoms, hopefully during primary infection, so that it can be easily treated. Okay, so we're looking at x-rays here of a person's thoracic cavity. We're looking at the rib cage here, intercostal bones, and right here is a tuberculosis lesion within the right side of the thoracic cavity, inside of the right lung. Okay, so that's one way to diagnose the disease. Another way is by a tuberculin skin test. Okay, so what they do, you probably have had it before. What they do is they insert, or they inject, antibodies right under the skin, so subcutaneously, and they usually do it on the arm. So if after a few days the little bump is gone, right, because it does form a bump that is visible on superficially on the skin, if it's gone within a few hours to a few days, then you're okay. But if it's not, if it gets bigger, larger, if it expands, then you probably have the infection. So they go on and you go on and get x-rays here, and that's what the doctor uses to diagnose people with tuberculosis. Alright, so antimicrobials are pretty ineffective, so the common antibiotics like penicillin, for example, so therefore combination therapy at high doses is going to be required, especially in advanced cases. There's also immunization, so there's a vaccine. So the BCG vaccine is administered to people, and I believe most people here should have the vaccine. But in preventing the disease, then you want to avoid inhaling respiratory droplets in patients that do have tuberculosis. So if you do know somebody who has tuberculosis, then keep your distance, if you will.